Okay, hello and welcome back to part 3 of Respiration. And so far we've done basic anatomy, we've looked at the purpose of the respiratory system in terms of bringing in oxygen and clearing carbon dioxide as a byproduct of the energy production in mitochondria inside the cells. In the last talk we looked at how we breathe, mechanically, how we use muscle contraction in the muscle sitting in between our rib cage and our diaphragm at the base of our thoracic cavity in order to increase volume inside the thoracic cavity. Because the lungs are attached onto the inside of the thoracic cavity due to hydrogen bonding in the serous fluid between the parietal and visceral pleural membrane, that the lungs also increase in volume. And from Boyle's law, we know that any increase in volume leads to a decrease in pressure. So once we decrease pressure below the pressure of the air around us, air flows in. So that whole contraction of the muscles expanding our chest, essentially, decreases the pressure inside the lungs and allows air to flow in down a concentration gradient. When we expire or exhalate, we just simply relax those muscles as they go back to their originating positions. The thoracic cage shrinks, the volume decreases, pressure increases above atmospheric pressure, and the air is pushed out. So that's basically how we do it. Now we need to think about how we control that. So if that's all that we did, then we'd just be set at one particular level of breathing and we wouldn't change that level of breathing. But if that were the case, we'd actually be quite limited in what we could do, because keep in mind, the more that we breathe, or the faster and deeper that we breathe, the more oxygen that we're potentially getting into our blood, sending through to the cells to be made into energy. So as we increase our activity levels, our breathing naturally will increase too. And of course, any time that we go to the gym or play sport, etc., we recognize that very quickly, that we find ourselves breathing more quickly and more heavily. So as we require more oxygen, we breathe more quickly and we breathe more deeply in order to supply that oxygen. Now, back when we looked at muscles, we looked at a little bit of sports science, so we realized that most of the time we actually are in oxygen debt. But that, if you're interested in that stuff, go back to that point and have a look at the PowerPoints, because it's included there. Or send me questions and I can answer them directly for you. Anyway, so how do we control that? So we start exercising, we need to breathe more. There's got to be a system in place for that. And since we've been studying physiology, you should hopefully be sitting there thinking, okay, well that's homeostasis, and homeostasis is controlled two ways, negative and positive feedbacks. Now, positive feedbacks tend to be quite, extreme things, so you're pushing away from a norm, until you get to a point, and then everything is fixed, like clotting of blood, for example. Well, needing oxygen isn't quite on that kind of scale, and of course we need to go back to normal breathing at some point later, but what we do have is a negative feedback system, and we know that both systems rely on stimulation. So first of all we have to think about, well, what's stimulating the process, and then receptors, and then how those receptors link to the control center, and then what comes back. So really what we're doing when we're thinking of extrinsic control of mechanical breathing and respiration, which is quite a long, complicated sounding sentence, what we're really thinking about is what is the negative feedback system that controls this, and you're just naming the parts. Well, what's the stimulator, or what's the receptor, what's the control center, what part does the control center play, and then what gets stimulated in turn down through the efferents, and then we get our change. So that's what we're going to look at now. So starting with that in mind, when we're looking at negative feedback, first of all we're thinking about, okay, well what's going to stimulate? Now we've got two possibles here. Now the obvious choice here is probably going to be oxygen, because that's the thing that we're going to need more of. So the more that we exercise, the more oxygen we need. Therefore, that would then indicate as oxygen levels drop in the blood, then we're going to be stimulated to breathe. Now, that actually isn't how we do this. Sounds surprising, but there's a reason for that. If we were along that system, a lot of other things could build up and become a problem. So we actually are sensitive to carbon dioxide levels in our blood. Now, at the end of the introduction, I showed you an equation of carbon dioxide and water and reminded you that carbon dioxide is a byproduct from the cellular respiration pathway, which produces ATP in the mitochondria. Carbon dioxide in water gives us carbonic acid, which breaks down into the hydrogen ion, which gives us a type of acidity, and also a bicarbonate, which is negatively charged. So, as we exercise, we need more energy. That's why we need more oxygen. But we also, as a result of that, produce more carbon dioxide. Now, the problem with that is that as carbon dioxide, is being created, well, it needs to be carried back to the lungs and literally blown off in our breath. We have sensors built into our system for that. Now, we have them both inside the blood and centrally inside the central nervous system. So how are they stimulated? Well, let's remind ourselves of the equation, if I can find somewhere to stick it. I'm going to stick it up here. CO2 plus water goes to H2CO3, and that can dissociate into hydrogen ion and bicarbonate. Alright, so as we start exercising, that goes up. That also goes up. So the equation gets pushed in that direction. So we start producing a lot of this. Now inside the cells, if that gets too high, then cellular chemistry starts being interrupted. So the hydrogen ions are got rid of, they're pushed into the blood, and then the blood starts becoming more acidic. Now, in the peripheral area, that is where the sensors are in the blood. So in the carotids, feeding, carotids either side here, feeding the brain itself, and the aortic bodies right off the heart. So as the major vessel is leaving the left side of the heart to going to the system, there's sensors in there. So as carbon dioxide increases, you get an increase in hydrogen ions, because as that goes up, that goes up. And so we have receptors for acidity in the carotids and the aortic bodies of the blood system, which then send that information to the brain. We also have a central area as well. So in the central part, we're talking about the central nervous system, and so we detect that in the cerebrospinal fluid, or CSF. Now this is an indirect stimulation, and it's indirect because, if you remember, we have a blood-brain barrier, meaning that when capillaries go through the brain, things do not leave the capillaries and through the gaps and just leak out and then just spread into the brain cells. They're actually sealed, and the only way to get something through a blood capillary that feeds the brain is going directly through the cell. It then is met on the other side by an astrocyte, which then filters that, and it doesn't get to the neurons until it's gone through all of that process. So a lot of things are actually prevented from getting there. One of those things is hydrogen ions. So in the periphery it's fine, but as this is going up here, that will not be going up necessarily or directly crossing into the cerebrospinal fluid. But what can cross is carbon dioxide, because it's a gas and it can cross the membranes. So in the central system, we're actually detecting carbon dioxide. Now it still goes through that process, and we're still essentially doing that. But we learn that in the central system, carbon dioxide, however much carbon dioxide crosses the blood-brain barrier, is what stimulates the response to the brain. And in the blood vessels themselves, it's the buildup of hydrogen ions that do it, or buildup of acidity. Okay, so this is how we detect it. So we've now named the stimulant, which is carbon dioxide. We've now named the receptors in the aorta and the carotids for the blood, and in the cerebrospinal fluid, receptors in the walls of that near the epidermal cells, in the central nervous system. So now, where are we sending it? We've got to send it to a control center. So the control center in this is actually the pons, which is a very primitive part of the brain, and the control center has two parts. It's got apneustic and a pneumotaxic. Now, at the 200 level, we keep this relatively simple. So the stimulation comes in as we're getting an increase in hydrogen ions going up and CO2 in the central parts. That stimulates the apneustic center to stimulate the rhythmicity center in the medulla oblongata. And that's what causes us to breathe faster. If we finish our exercising and we're calming down and slowing down, and we're basically now got enough energy to do our cellular chemistry, we don't need to breathe as fast, the pneumotaxic area inhibits the stimulation from the apneustic and the breathing slows down. So these two antagonize each other. Apneustic stimulates faster breathing, the pneumotaxic slows it down. So big arrow up for apneustic, big arrow down for pneumotaxic. So this is our extrinsic control. We've now got to the control center. But we need to actually then, from the control center, get it to the effector. So the effector in this case is really kind of in two parts. The first part is up here, still in the brain. So we've gone from the central nervous system, cerebrospinal fluid, for example, to the pons in the brain, and then to the medulla oblongata directly below it. And this is the simplest part of the brain altogether. And what we have here is a basic rhythmicity center. So if someone comes up behind you and basically goes bonk on the back of the head, and then you pass out on the floor, you don't, well, unless it's very severe, you don't stop breathing. Or if your 2-year-old child decides that they want chocolate cake and ice cream, and you say, no, you can't have chocolate cake and ice cream, and they do that really cute thing where they hold their breath until they pass out, which, I'm being sarcastic, is not cute, it's quite stressful, but it's one of those battles that you have to fight sometimes. They don't just stop breathing on the floor, they start breathing again. And this is because of the rhythmicity center in the medulla oblongata. Just sits in the background, keeping everything going. Just kind of like a drum beat. The drummer's always there keeping the rhythm going so everyone else can stay on time. Now, in the rhythmicity center, we've got two neuron areas, the DRG, etc., and VARG, which you can look up in the PowerPoint if you're interested. The I neurons are so named because they are for inhalation, the E neurons for exhalation. So what's happening here is that, at rest, the I neurons are being stimulated, and they send the stimulus down through the spinal nerves and the phrenic nerve, which feeds the diaphragm and causes the contraction in the intercostal muscles and the contraction in the diaphragm, which we talked about in the last one, which increases the volume, causes the chest to come out and up, and the diaphragm to go down. Increase in volume, decrease in pressure, in comes the air. So that's coming from the medulla oblongata. We don't inhale all the time, we exhale, and that's the E neurons which are inhibiting that signal. So then we get relaxation of the muscles and everything comes back to normal. And then we get a new stimulation from I neurons and off we go again. Now, at rest, we're normally breathing anywhere between 12 breaths per minute to 18 breaths per minute. Most of us will probably be sitting around 12 to 14 when we're not doing too much. And so we get a change. Suddenly, the dog's just jumped over the fence, is now chasing a car down the road, and you've got to get up and get the dog back. So you're, out your armchair, you're rushing off down. So that's picked up here in the periphery and in the central. So hydrogen ions in the periphery, building up in the blood, coming from the cells. Remember, through this thing here, this equation here, and in this the central area, because CO2 is crossed into the fluid of the cerebrospinal fluid, and then stimulated. So these two are now stimulating hard the apneustic center, and the apneustic center stimulates the eye neurons more regularly. So they're now stimulating the contraction of the chest more regularly. So you've gone from to, so you're now getting more stimulation coming through the eye neurons. Once you've caught the dog, and you and dog have got back to your house and everything's calm again, and you're sitting back in your armchair, you'll get inhibition from pneumotaxic, because this will have dropped down. The hydrogen ions will have dropped down. The CO2 crossing the central barrier will have dropped down. We're now moving more in that direction and away from building all this up. So the pneumotaxic inhibits this signal, so the eye neurons are told to fire less frequently, and you breathe more slowly and more regularly. That is how we extrinsically control our breathing. So remember, just always kind of think of it, think of it straight, negative feedback. What is our stimulator? What's stimulating? What receptors do we have? Where's the control center? And where's the effector that comes off that control center? So hopefully you've now got an idea of how that works in the lungs. Alright, so that's it for this one, and when we come back we'll talk about gaseous exchange and transport.